Transpiration is caused by the evaporation and diffusion of water from the plant's surface, and mostly occurs through leaves through stomata, which are small pores found at the bottom of them. This process helps to draw water up from the roots and through the plant. The route that the water takes is known as the transpiration stream, and it starts off in the root hair cells where water is taken in by osmosis, then goes up the xylem and out of the stomata in the leaves where water evaporates. This movement of water is a key part of the plant's water management and nutrient distribution. The rate of transpiration is affected by 4 different factors. The first is light intensity. The greater the light intensity shining on a plant, the faster the rate of transpiration. This is because the stomata open up in bright conditions to allow more gas exchange for photosynthesis. As a consequence of this, more water gets lost through transpiration. The second factor is temperature, where the higher the temperature, the faster the rate of transpiration. This is because energy of the water molecules increases, which makes them move faster. This means that the rate of diffusion and evaporation of the water molecules out of the stomata increases. The third factor is air flow rate, which is basically the wind speed around the plant. The higher the air flow rate, the faster the transpiration rate. This is because air removes water vapor from the surface of the leaf and keeps the concentration of the water outside the leaf lower than the inside. This increase in concentration gradient makes diffusion faster. The fourth factor is humidity, which is basically the amount of water vapor that there is in the air. The higher the humidity, the lower the transpiration rate. This is because it means that there's a high water concentration outside of the leaf, which results in a low concentration gradient, which results in a slower diffusion. You can measure the rate of transpiration by using a potometer to measure water uptake by using the following setup. Just fill the potometer with water and mark the starting position of an air bubble in the capillary tube. Begin the experiment by starting a stopwatch to measure the time, and you'll notice the air bubble move as the plant uptakes water. After a set amount of time, you can note the distance moved by the air bubble to calculate the rate of water uptake. This will give you an estimate of the rate of transpiration, as you can assume that the amount of water taken in by the plant is the same as the water lost in the leaves. So that means the greater the distance that the air bubble moves in a period of time, the faster the rate of transpiration. Okay, now let's try a quiz on this topic. See how many questions you can get right. If you liked that video and want to see more, remember to subscribe to Flash Revision Lab to get notified every time we release new content.